Alrighty guys, and we are back here looking at section 2, more particular about our genetic crosses. So here I do have an example of a Punnett square up here, going back to those pea plants that we were talking about before, and I'm also showing you alleles as well. So I have my female plant going down here on the one side, my male plant is up here above, mom and dad plants. You can see what their alleles were. They're both heterozygous, so we have a capital B and a lowercase b. And dad also has a capital B and a lowercase b. Whenever you cross those two together, it'll show you the potential for the children, what they could end up being. I'll explain that more in detail as we go through this. So let's start off with some vocab terms. Genotype. These are the alleles that an organism inherits from the parents. They can be dominant, recessive. You get one from each parent. So for purple, we're going to have two capital B's. If you have a capital lowercase b, it's still going to be the color purple. If you have two lowercase b's, that is going to give you the color white. The letters down here are representing our genotype. Couple prefixes that we want to pay attention to. We have homeo or homo, which both mean same, and hetero, which means different. There are three different possibilities for genotypes when two alleles are present, and we're just going to be sticking with the two allele thing for the purpose of this class. If you were to take an upper level biology class or even a genetics class, we would introduce more letters. So if you have two capital letters, we are going to call that genotype homozygous, homo because they are both capital, homozygous dominant. If you have two lowercase letters here, it's going to be homozygous because they are both the same, and recessive because they are lowercase. If you have one of each, a capital and a lowercase, that is going to be heterozygous. Hetero means that they are different. Phenotype is an organism's appearance, so this is what you can see. You can't see somebody's genes. You can't see what their genes for their hair color are or their eye color, but you can tell if they're blonde or if they have brown eyes. So again, we have our two capital B's. This is our genotype. The color is going to be our phenotype. Big and a little b, going to give you a phenotype that's purple. Two little b's is going to give you a phenotype that is white. Probability, we'll be doing that a lot as well, and if you need a calculator, I'll give you one, but it's fairly simple. You have like 4 options. If I count 4 options for your probability. You could either have a 0% chance, a 25% chance, 50% chance, or a 100% chance. Oops. There are 5. And 75% chance. So it's a likelihood that a specific event will occur. So probability, you're going to take the number of times an event occurred over the total number of events. For example, in Mendel's experiment, he counted the colors of pea pods and found that there were 6,022 pea pods that were green and 2,001 were yellow. What is the probability that the dominant trait will appear in a similar cross? So step 1, we're going to calculate the number of events here. So our total number of events, we're going to take 6,022 plus 2,001, and we're going to get 8,023 total events. Then we need to calculate our probability, which would be green pods over the total number of pods. So we had 6,022 green pods, and we calculated our total was going to be 8,023. That is going to give us 0.75. To express probability as a percentage, you take your decimal and you multiply it by 100. Or you're going to move your decimal place 2 points to the right. So 0.75 times 100 would give you 75%. Example number 2. In Mendel's experiment, he counted height of the pea plants and found that 787 were tall and 277 were short. what is the probability that a recessive trait will appear in a similar cross? So we need to calculate the total number of events. So we take 787 plus 277, because those were the two numbers we were given. We know that this many were tall and these ones were short, so we have to add them together to see the total number of plants that we had, which was 1,064. So we're going to take short plants, because that is what we are looking at right now, divided by our total plants. So you take 277 divided by 1,064, and that gives you 0.26. Express that as a percentage. You're going to multiply it by 100, or move that decimal 2 places to the right, and that is going to give you 26%. A monohybrid cross is a cross in which only one characteristic is going to be tracked. We tend to call these monohybrids. It's just the offspring of a monohybrid cross. And we're going to be using Punnett squares, which is a diagram used by biologists to aid in predicting the distribution of inherited traits by the offspring. So flower color is passed through complete dominance. Purple flowers are dominant to white flowers. Cross a homozygous dominant flower with a homozygous recessive flower. So here's my two flowers. My dominant flower we know is homozygous, which means that they are both going to be the same. And since it's dominant, we're going to make them capital. For my white flower, it is homozygous, so they're both going to be the same. Since it is recessive, they're going to be lowercase. When you make a Punnett square, it is very important that you keep the alleles for one parent together. So notice how both purple alleles went on top. You could, if you wanted to, put both purple alleles on the side. You cannot put one purple allele up top and one on the side. They either both go on top or both go on the side. Only two options that you have. My white flower alleles are going to go to the side, since that's my open spot. And then what you do is you're going to pull down and across, and in each one of these boxes, you're going to end up with two letters. That's going to give us a new genotype. So I slid my capital R down, move my lowercase r over, and there are my offspring. Genotypic ratio is the ratio of genotypes that appear in the offspring. When we write our ratios, we're going to go homozygous dominant, heterozygous, homozygous recessive. Phenotypic ratio is just the ratio of the offspring's phenotypes. So normally it's going to be some type of color. So what is the genotypic and phenotypic ratios of our offspring? So that was our Punnett square. When we do our genotypic ratio, we're doing homozygous dominant first, and you have to write it even if you don't have it. So 2 capital R's. There are no 2 capital R's in here, that's why that's a 0. Heterozygous, 1 capital, 1 little. All 4 blocks are like that, so you have 4. And we're looking for homozygous recessive, which there are none, so it is a 0 again. Make sure you have your colons that separate it. That's how you write a ratio. When you do your phenotypic ratio, you do dominant first and then recessive second. So purple. All these flowers are going to be purple because they have at least 1 capital letter in here. We're not going to have any white flowers. There are other forms of inheritance other than complete dominance. What I just showed you was complete dominance. And we are going to be practicing complete dominance next week. Then we're going to take a stab at incomplete dominance, and then we'll do codominance. All have the same basic concept, so once you know how to do a Punnett square, you'll be fine. It's just remembering which type of dominance you're doing. So I'm going to show you examples of the other 2 types of dominance now, just so you have them written down and you're ready to go. Which I would suggest that you maybe keep a little notebook and write these down in it, so that whenever you are doing problems on your own, you have that to refer back to. So incomplete dominance is a type of inheritance where heterozygous genotypes are going to be blending. That means that there is no actual dominant trait. They just mesh together. For example, if 2 capital B's gives you blue flowers and 2 lowercase b's gives you yellow flowers, then whenever you get a heterozygous, you're going to mix and get green. Black wool sheep are dominant to white wool sheep. The color of the wool is inherited by incomplete dominance. What would be the color of the heterozygous offspring of a black and white sheep? So here we have a black sheep, here we have a white sheep. And you would probably end up with some type of a gray sheep. So examples. Red flowers are dominant to white flowers, and most of the time, at least when we're starting off, I will tell you which one is dominant and which one is recessive. They are inherited by incomplete dominance, so we already know here that we're going to have a blending of the traits. So that means that my heterozygous is going to be pink. Red plus white would give me pink. What would be the genotypic and phenotypic ratio of the offspring of a flower that is homozygous recessive and a flower that is heterozygous? So we have homozygous recessive for one flower, heterozygous for the other flower. My homozygous recessive genotype is going to be 2 lowercase r's. Homo meaning they're the same, recessive tells me that they are little. Heterozygous is going to be 1 of each. Hetero means they are different, and that's pink. When you talk about heterozygotes, you don't have to say heterozygous dominant or heterozygous recessive because there is no such thing. It is just heterozygous on its own. The only time you refer to that as a homozygous whenever you do have to differentiate whether it is recessive or dominant. All right, so we're going to put in our alleles here. So my homozygous recessive, I'm going to put those ones on top. And my heterozygous ones, I'll throw those on the side. And then you're going to be pushing things across and bringing them down. Whenever you write your Punnett squares, if there is a capital letter, you always want to write that one first because it's easier to read that way. You won't get yourself confused. So genotypic ratio. Do we have any that are homozygous dominant? That's 2 capital R's. Look in here. We do not. Heterozygous. That's 1 of each. We have 2 that are heterozygous. And homozygous recessive, that's going to be down here. We have 2 that are homozygous recessive. So there would be our genotypic ratio, 0 to 2 to 2. For our phenotypic ratio, we are now going to have 3 different colors. We'll have red, we'll have pink, and we'll have white. When we have regular complete dominance, then you only have 2 colors. But with incomplete dominance, you do have 3 because you have that blending happening. So, in order to have a red flower, we have to have 2 capital R's, which we don't have, so there aren't any red flowers. Pink flowers are heterozygous, so we're going to have 2 pink flowers, and white flowers are going to be homozygous recessive. We have 2 of those. When you do these ratios, they should match. So your genotypic and phenotypic ratio, if you notice, it's 0, 2, and 2. When you have incomplete dominance, that's a very easy way to check yourself. Second example, red flowers are dominant to white flowers. It is inherited by incomplete dominance. What would be the genotypic and phenotypic ratio of the offspring of a flower that is homozygous recessive with a flower that is homozygous dominant? Okay, so homozygous recessive is going to be 2 little ones. That's going to give us a white flower. Homozygous dominant is going to be 2 capital letters. That's going to give us a red flower. Whenever I do my mix, make sure you keep your parents together. There we go. Genotypic ratio. We don't have any that are 2 capital letters. We have 4 that are heterozygous. And we have none that are homozygous recessive. For the phenotypic ratio, we have no red ones, we have 4 pink ones, and we have no white ones. Again, our ratios should match. That means we are correct. The second type of dominance is codominance. And this type of inheritance is where both alleles for a gene are going to be expressed in the heterozygous form. So they are both dominant. They fight with each other. We'll use 2 different letters for codominance. This is because both traits are expressed and no one is more dominant than the other one. For example, red color on horses is going to be 2 capital R's, white color on horses is 2 capital W's. So we have 2 different letters now because we don't have a recessive form. And if you were to use a lowercase letter, then in your mind you would be thinking, oh, that's recessive. The heterozygous is going to be RW, which is going to give you a roan horse, which is a horse that has white coloring with red spots. Example 1, horse color is inherited by codominance. When a red and white horse mate, the offspring has a white base color with red spots. This is called a roan horse. What would be the genotypic and phenotypic ratio of these horses? So there's my red horse, there's my roan horse. When I do my copies, keep my parents together. Genotypic ratio, you start with your R's, your RWs, your WWs. And phenotypic ratio, we're going to have red, roan, and then white. Another example of codominance is blood type. So a person's blood type is made up of the alleles A, B, and O. The alleles A and B are equally dominant, however, O acts as a recessive when you compare it to A and B. So a person with A blood type could either have the genotype AA or AO. People with A blood can only receive blood that is either A or blood type O. A person with blood B has either BB or BO. People with B blood can only receive blood from B or O people. A person with AB blood can only have one genotype, and that is AB, because you only have 2 alleles. It's the universal recipient because they can receive blood from anybody. And a person with O blood can also only have one genotype, which is OO. They're called the universal donor because they can donate to anybody, but they can only receive from people that are O. O is generally speaking going to be the recessive form of blood types. We'll do a whole day or two just talking about blood types, so I'm not going to harp too much on this stuff right now. This is just showing you donor versus recipient. We'll do an example here. We have a husband has O blood and his wife is AB. What are the genotypic and phenotypic ratios of their potential offspring? So the husband only has one genotype and the wife only has one genotype. When we mix them together, genotypic ratio, we're going to have 2 AOs, 2 BOs. Phenotypic ratio, we have 2 that are A, 2 that are B. The O kind of goes away, it's silent, it's hidden. With genotype, because there are so many different potentials of blood, you only have to write down what you have. We'll do one more, I think. The husband is heterozygous for A, his wife is heterozygous for B. Genotypic and phenotypic ratios. So if you know they're heterozygous, that means they're going to be different. So AO and BO. Keep your wife up top, keep your husband up top, keep your wife on the side. And they could have a mashup of different kids. They could have AB, they could have AO, BO, or OO. Phenotypically speaking, we could have AB, A, B, or O for children. Didn't want to confuse you too much with the blood typing stuff. Like I said, we'll spend a lot of time just talking about blood type, but that is it for today.